Alrighty, so today we've got Dr. Ronald Blanton. He is the chair of tropical medicine at Tulane. I'm very excited to have him participate in our speaker series this week for National Public Health Week. All of us know, right now is unfortunately a great time to be involved in the field of public health. Lots going on with the pandemic and beyond. But today, Dr. Blanton is going to share a bit about his journey in public health, maybe some research that he's worked on, and hopefully many of you watching, if you're on the fence about public health or what area to focus in, or maybe you're just watching because you're interested in the topic, I think you're going to be able to learn a lot in this over this next hour. So you can go ahead and take over.
Hi, welcome everyone. I'm, as advertised, Ronald Blanton. I'm the chairman of the Department of Tropical Medicine here at the Tulane School of Public Health and Tropical Medicine. Our department is in the name. So I think it is one of the foundational departments of the whole school. It's one of the reasons for us to be here. If I can blow at least the horn of the department a bit. Let me share my screen. A second. Is that up there? Yes. Is that up for everyone?
I can see it.
Okay. So I was tasked with talking about at least my journey into global health and to impart what it takes. And after we get rid of that part, I'd like to talk a little bit about what I do, what interests me in this. That is one area that I have in terms of research. What I want to show you initially is essentially this is the career path for global health. If you want a career in global health, you're looking at it, which is there is no career path. And this is not unusual. Unless you're going into a professional school, medicine, law, accounting, nursing, a veterinary school, where there is a clear curriculum in which you're going to make it, most people in most jobs are going to have to find their way based on what they know and what the opportunities that present themselves to them. And it's no different in global health, I don't think. I didn't say this, I wish I had. But a father once went home to his son and asked him, well, what are you going to do when you grow up? And he said, Dad, why do you ask such a stupid question? The job I'm going to do has not even been invented yet, which was just prescient. I think that was really true, especially now. You don't know what you're going to encounter. There are some guides that perhaps I can give you. And again, this is my career path in global health, and it may not be yours. There are many. First of all, I suggest you make yourself big. And what I mean by that is to develop a whole wide range of interest in a lot of different things. You don't have to be an expert in anything, but there's no reason not to have an interest in astrophysics at the same time that you like hip hop and classical music. That all of these things will eventually end up being useful in terms of whatever you do, but certainly in global health, which is a multidisciplinary area, all of this comes into play. While you're making yourself big, you should also be good at something. That is, there should be something that you're known for that gets you in the door, some skill that you have or some knowledge that you have. You should develop a wide range of skills. That, again, is not to say that you can do ELISAs or matter of fact, I've never done ELISA in my life. But you should develop a range of skills and interest in different areas and different things, because you never know where the opportunities will lie, that you'll have to take advantage of those things. And while you're making yourself big and making yourself small, you should also develop a differentiating skill. That is something that you are best at that other people are not, that somebody wants, that is something that's sellable. Finally, you should learn a language, and that includes English. Any language will do. If you're in South London, Urdu may be the language for you. If you know Arabic, you could be dumb as dirt, and nowadays people will love you, they will need you, there'll be a place for you. But you should also learn English well. Anybody who is under the impression that I spend my time in front of a PCR machine is greatly mistaken. Most of the time, what investigators do, whether they are at any level, is sit in front of a computer. Most of it is analysis, 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 reading and writing. And if you can write well, if you can tell a good story, not a good joke, but a good story with a beginning, a middle, and an end that has some importance for people, that's going to get you far in this profession or in this area of work. My own personal career begins before 1970, which is, I have no idea, well, certainly at that time, where I was going or what I was going to do. But as it turns out, for reasons that are still not clear to me, I took Spanish from the 3rd grade on at the public school system in Omaha, Nebraska. That shows you how times have changed. This was totally free. I have no reason to, no idea why I was doing that. I didn't know anybody who spoke Spanish. I can only think that there were not computer games and my mother wanted me out of the house. So it became a theme. It's something that I pulled on, even though at the time I had no idea what I was doing with this. In 1970, I went to college. I majored in English literature, which you could in those days, and still go to medical school. I also did Spanish literature. In 1974, before I went to medical school, I got a grant to study folk medicine on the north coast of Peru. Again, there was no good reason for me to do that. It was suggested by a girlfriend. I still work in South America. I no longer have the girlfriend. But these things come from places that you may least expect it. In 1975 to 79, I went to medical school at Case Western Reserve University. Did internal medicine at University of Illinois in Chicago, which was a great experience. It also was a great international experience. There is a pipeline straight from Mexico City. If you go to the bus station there and look on the maps, there is a red line that goes directly from Mexico City right up to Chicago. And a lot of the people that I saw in the hospital were actually immigrants from Mexico. And we saw a lot of diseases that you see in Mexico and not necessarily in Illinois. Well, you do see them in Illinois because of immigration, but internal medicine residency in Chicago. In 1981 to 85, I came back and did a fellowship in internal medicine, fully expecting to go work somewhere in South America in some small little clinic, because I got a great deal of satisfaction from seeing patients. And I still do, but as it turns out, part of the fellowship, you had to do research, and I discovered something that surprised me, which is that I actually had an interest, that I actually enjoyed research, which is different than studying biochemistry in college or studying physics or anything else. It takes a different muscle. And people who are very good at one are not necessarily good at the other. I know a fair number of English majors who've ended up in basic research. So this should not be so surprising. But it surprised me that I was interested in it, because as far as I could tell, the reason for research is so that you could take those people who are socially inept and put them somewhere. Hopefully that's not the case. But I found that I was interested in having questions that I could devise and answers that I could receive. And they're not big questions. Sometimes they're just a band on a gel was very satisfactory to me. I had an apprenticeship in molecular biology. The only reason they put me in this is because the person who had been in that position before decided to go to Alaska. And so they said, Blanton can do this. So they put me in that position, which I thought was totally out of space for a guy who comes out of English and internal medicine. But as it turns out, molecular biology is really a discipline that is fairly conceptually simple. It's difficult to do, but conceptually it's fairly simple. And that was a great experience to be exposed to people who are more focused on basic research than on clinical medicine. And there is a different attitude in the way that people approach problems and data that I think was invaluable from that experience. But again, I did not plan to go into molecular biology. I eventually was on the faculty at Case Western Reserve and did a lot of research in various places, Egypt, Kenya, and Brazil principally. In 2000, there was a key change, which is I was forced, because a colleague of mine left the program at Case Western, to take up genetic epidemiology. I had to go back to school. And that made all the difference in terms of my career, not necessarily making it any better, but making my satisfaction in terms of what I was doing and making understanding science a lot better. And finally, just maybe 2 years ago, less than 2 years ago, I became the chairman of the department here. Getting back to this master's in genetic epidemiology, what was important for it is, first of all, math for dummies. For the epidemiology program at Case Western at that time, they gave you a list of mathematical problems to solve to see how far along you were and whether you needed any help. And as far as I was concerned, I had found for 30 years I had no use for calculus. I thought I'd finally found the one thing that was useless in my life. But during this course in Math for Dummies, I was shocked to find that everything that fell out of the professor's mouth was actually relevant to things that I was doing, that I could see real epidemiology. By that, I mean, not just the registering of people and places, but actually designing things so that I had a decent answer to a question that I was asking. And genetic epidemiology, which wasn't such a stretch since I was already involved with DNA was not a foreign concept to me, and working with DNA was not foreign. And genetic epidemiology and epidemiology seemed to mesh very well in that. But what I mean by being able to change my career is that you begin to realize that what science is really seeing better. That if I, for instance, asked you to go down Canal Street here, one of the main drags in the center of the business district in New Orleans, and count every clothing store along the way on the uptown side of the street, you would see I don't care actually, but you would see every clothing store on the uptown side of the street, but you'd also see all of the stores that are not clothing stores. You'd see a lot more than if you just walk down the street. When Isaac Newton says that he could see further, he's just talking about science. Science is being a nearsighted guy who puts on his glasses to cross the street. So to be able to see better, always had this image in my mind, is the Spiderwick Chronicles, that if you just put on a pair of glasses, you'll see things that are there that were imperceptible prior to actually doing that kind of analysis. And I think that's what I got from this discipline of looking at genetic epidemiology and combining that with what I do in laboratory science. The problems that I ended up working on and the tools that I used to work with, I worked on various problems. And in part, that is because of the discipline I am in. It is hard to do just one thing. So the opportunities came up and I took them to work in schistosomiasis, which is the principal area I've worked. But I've also done significant studies on echinococcosis, hepatitis C, dengue, lymphatic filariasis, and antibiotic resistance. May not seem like a lot of relationship between all of these, but there actually is, if nothing else, in terms of place. That is, I was someplace where nobody was doing the study and there was good questions that could be asked in these areas. In addition, you have to develop a whole set of tools as well if you're working in these areas. And the things that I've done, human biology comes directly from working in medicine, although you don't have to be a doctor to have the importance of human biology being, in essence, usually the diseases we study are diseases of people. Molecular biology, of course, is where I sort of started, at least in terms of basic science. Immunology, I hate immunology, but I have to use it. Epidemiology, genetics, and English literature. Again, nothing ends up being wasted. And the entire time since I was in the 5th grade, I've been able to pull on all of the things that I have actually been exposed to. So the disease, to stop talking about me, that has occupied most of my time, but not all of it, is a disease called schistosomiasis. This is a disease in which there are 3 main species that affect humans. It's displayed here in what we call a 2 boys slide, because it seems like we always trot out the same slide of these 2 boys to talk about schistosomiasis. And the idea is that both of these boys are infected, but obviously only one of them is really sick with the disease. There are over 250 million people infected with schistosomiasis, and only about 10% of them end up with severe disease. Only 10% means 25 million people, or 5% means 13 million people who are going to end up in this condition, which is portal hypertension is the main severe morbidity associated with schistosomiasis mansoni, in which the portal vein ends up being blocked. It sends the blood up the splenic vein that opens up collaterals in a whole lot of different areas, like you see this large vein across the abdomen of this boy. In addition to that, he has varices in his esophagus. And those varices, unlike the ones on people's legs, are not protected by a thick layer of skin. In fact, there's only a thin layer of tissue between you and that blood vessel or that fishbone, and people die by bleeding out through the esophagus. The other boy is also infected with schistosomiasis, but you can't see his morbidity. Even mild and light infections with this parasite are associated with growth stunting and anemia. But again, they are considered lesser morbidities, although they may have a bigger impact since 90% of the populations, that's 250 million people, may suffer from that. You may notice also that the boy who clearly has portal hypertension also suffers from malnutrition with, you can see all of his ribs along here. He has temporal wasting along here, wasting at the shoulders. So he's a severely malnourished young man. So this is a disease that's present throughout the tropics. Even though we're in the subtropics here in New Orleans, it is not a disease we see in New Orleans or even Florida, but it used to be a common disease in Puerto Rico, for instance, and through other islands in the Caribbean. It is transmitted by a freshwater snail, and it transmits by direct penetration through the skin. It is a dioecious parasite, which means there are morphologically different male and female worms. And what you see here below is not a single worm, it's actually the pair. It's a pair of male, which is the large worm with these projections on the surface. And the female is this smoother round worm that fits inside a canal that's in the body of the male worm. And in fact, that's the name. Schistosome means split body. And that split body is because the female resides within the gynecophoric canal. And these parasites exist in a state of constant copula. So you can see that there is romance in parasitology, no matter what anybody says. That these two are rounded up, even though they're flatworms, because they live in the mesenteric veins. They live inside of veins, so they take that shape. The females will begin to lay eggs, 300 to 3,000 a day per worm. And those eggs, some of them will go out in the stool or in the urine, depending on the species, but some of them end up being swept back into the liver and cause this disease. The disease itself is a result of the immune response to parasite eggs in the liver or in the bladder in the case of hematobium, Schistosoma hematobium, which is around the bladder. And it causes portal hypertension in the case of Schistosoma mansoni and Schistosoma japonicum. The treatment is praziquantel. It's a drug, it's a pill. They're 80 to 90% effective. It's a pill that costs about 8 cents a pill, and 6 pills will treat an adult. So we're talking all of 50 cents in order to treat somebody, which is really inexpensive in most places. But what happens is that there is not sterile immunity to schistosomes, so that even after treatment, reinfection is extremely common. So that the recommendation by the WHO is that in places where there is a high prevalence of schistosomiasis, population should be treated every 1 to 2 years. If you were a poor country, although the drug is cheap, going to treat your entire population every 1 to 2 years is a very expensive proposition. What are alternatives? Why can't we eliminate schistosomiasis, since it's 80 to 90% effective, by simple repeated treatments? And one approach to this is just to go and do straight up shoe leather epidemiology and look at the number of people who are infected year to year and do an estimate of that. But I don't think that is really adequate to understand why you need to repeat infections. One way to begin to understand that is to take a conservation genetics point of view from the point of view of the worm. That is, what has happened with parasite populations that allow them to persist. So what we try to do is to put population genetics at the service, at the service of public health, to answer these kinds of research questions. What are the reasons for the parasite to exist in certain places where we don't expect it, like in urban areas? What are the reasons for it to persist despite repeated treatments? What does extinction genetically look like? What we're really talking about when we're trying to eliminate or eradicate parasites is essentially extinction of that species. Normally, we're trying not to avoid extinction, but that's not the case when we talk about diseases of humans. Is there an objective stopping rule? Even if we can't bring this parasite to extinction, is there a way we can tell communities or countries that you need to treat up until this point and the population will collapse on its own, or you need to treat up until this point and you only need to return every 5 or 10 years instead of every other year? Those may be questions that can be answered by specific indices that we see in population genetics. Population genetics is an area that's used a lot in conservation, as people may be familiar with it, to trace movements, to monitor the efficacy of control measures, to plan interventions, to determine the distribution of diversity of organisms. and to predict and to promote extinction. This is the thing that's really wonderful about it, is that it tells us about the past of a population. We can look at the present of a population, and we can predict. We have a crystal ball. We can kind of see the future of a population with some, not perfectly, but to some extent using these approaches. Population genetics, I will tell you, is a dismal science, perhaps as dismal as they say economics is, in that it is, if you're not a mathematician, because it is mathematically intensive. But there are two areas that really interest me in terms of population genetics and, in terms of public health, and that can be applied to public health, that simplify some of the problems. I'm not really interested with which worm is sleeping with which worm. I'm more interested in differentiation of the entire population. Is this population different from that population geographically? Is this population differentiated from the population that it was yesterday? And diversity, because diversity is the key to the persistence of populations. And those have certain measures. JOS-D, people may be familiar with the FST, is a measure that we use for differentiation. And the effective population size, or NE, is what we use to measure diversity. JOS-D or the FST, the idea is if you have two subpopulations within a large population, and maybe they're separated by a river or a road, and if you see that this population has sort of the same genetic characteristics of this population, that's a JOS-D of 0. There is no differentiation between them. They are genetically identical populations. Not individuals, but the populations are identical. And if, on the other hand, there's a road between them, a river between them, and there are no similarities between this population and the other population, that's a JOS-D or an FST of 1. So it's a number that ranges between 0 and 1. And there are guidelines for what that means. Generally, it's taken that JOS-D or an FST of 0.05 is negligible differentiation. 0.05 to 0.15 is considered moderate. 0.15 to 0.25 is considered great differentiation, and greater than 0.25 is very great differentiation. So we have some guidelines, some numbers to think about the populations and how they compare to each other. The other concept is a little more complex, but it's much more important in a way in populations, is the idea of effective population size. If we look at these two populations, there are 30 individuals in each population, the little round circles. You may not recognize them as parasites, but all of those are populations. They're individual parasites. And if the census number is 30, that's the number of individuals that we see in these two populations, and they are identical. However, if we were to do genotyping on these populations, we might see differences. That is, one population is more diverse than the other. One population, there are census of 30, but an effective population size of 2. Effective population size, one interpretation is that it's the minimum number of individuals necessary to recreate the diversity in a real population. So if we had a population of 1 million clones, the effective population size is, everybody say together, it's 1. Because we only need 1 individual to reproduce the diversity in a population of clones. In this second population, where there are 30 individuals, however, we say we need 12 individuals. It's not an exact number, really, it's less than that. 12 individuals to recompose the diversity in this population. If, for example, there is some change, or even if there is just genetic drift, that is random changes in transmission of genes between populations, just genetic drift or exposure to salt or heat or virus, something that the gray ones are sensitive to, our populations now look like this. And the ability of population 1 to resist that environmental change is much less than population 2. So effective population size really talks about the resiliency of a population. Let's go on a trip. Let's go to Brazil. It actually is probably, no, it's not a good idea. Brazil is having a terrible time with COVID right now. Don't go to Brazil right now. But if we were to go to Brazil, realize that it is the largest country in South America. It doesn't outline it well here, but all of this is Brazil. It is the size of the continental United States, and people don't realize this, but there are more people in South America that speak Portuguese than speak Spanish. It is a huge country. It is also a middle-income country. There are very poor people there, but there are very rich people, and there is somewhat of a middle class there. But it is richer than some Eastern European countries or even Western European countries that I could name. In South America, I'd say it is perhaps behind Chile, but probably on a par with Argentina and probably above economically most of the other countries in South America. It is the major place for schistosomiasis, however, in South America. And one of our objectives in studying Brazil for the moment is to understand why repeated treatments can or cannot eliminate schistosomiasis. And I'll give you some suggestions there. It might be immigration. You treat one place and all the parasites immigrate in from elsewhere. It might be poor coverage. We think we're treating everybody, but maybe half the people don't want to be treated and they don't get covered and they end up continuing the cycle for the parasite. Drug resistance. Praziquantel has been used since the 1970s. And some people think that there is resistance. Maybe resistance is why we can't eliminate this parasite. That there is poor detection. The methods that we use are mostly microscopic, and they may miss some of the people that are infected because they are not as sensitive as, they may not be the most sensitive method. There's some question as to whether other methods are really more sensitive, but still, we may miss a lot because of our method of detection. And sanitation is in parentheses because that's always a solution. If we had sanitation, we wouldn't even be talking here. So what we want to do is to define the problem in a small, isolated population. The reason for that is that I want to get everybody, I want to know where the community starts and where it ends, and I want to be able to touch and treat everybody in that community if we're going to try to eliminate it in this place. We want to do stool examinations 3 times for all of the residents. 3 times because a single stool is probably about 60% effective. 3 stool is only 80%. That means we're still going to be missing some people, but still 3 stools is better than just doing a single stool. And then we want to treat everybody that we find the first time through, come back 4 to 6 weeks later and re-examine everyone. The reason for that is that the parasite is initially insensitive to the drug until it's developed for about 4 weeks. So if I treat you with praziquantel today and you got infected 3 weeks ago, your parasites are going to survive that treatment. So we want to come back 3 weeks later and catch those parasites and treat everybody. And then we want to treat the entire community or go through about every 2 years, repeat this whole process. The place that we are talking about, again, this is Brazil, and this is the state of Bahia. It sort of looks like the map of Brazil. And right on the coast is this metropolis of Salvador. Salvador is the colonial capital of Brazil, and if I had more time, I could wax poetic about Salvador in terms of Bahia and what it means in terms of the culture of Brazil. Personally, much of what people think about being Brazilian, except their soccer teams are not the greatest, think about Brazil, it comes from Salvador, it comes from the state of Bahia. But we're going to leave that aside for now. And Salvador is a city of 3 million people. About 200 kilometers from there is a two small communities, Genipapo and Volta do Rio. Those communities are sandwiched between 2 larger communities of about 10,000. Over here is Santa Inês, and here is Ubaíra. And Volta do Rio and Genipapo are small villages along this Jiquiriçá River. They are separated by about 6 kilometers here and about 8 to 12 kilometers from the larger metropolis. But again, they are discrete. I know where they start and where they end. Genipapo has about 450 individuals. Volta do Rio has about 300 individuals. So I can census and treat everyone in this community. And this is the pattern of treatment that we did. Unfortunately, something happened in 2011, so we weren't able to repeat it there. But other than that, every other year, more or less, we have surveyed, treated, resurveyed, and retreated everyone in the community. What you may notice, I don't have it here. In the first year, there were about 10% of the population which was persistently positive that had to be retreated. But after that, we saw maybe 1 or 2 people that were still positive, but it was uncommon to see somebody who didn't respond to the very first treatment. Even though 10 to 20% of people is normal to not see people respond. And this is what we do for egg isolation. If you are a good parasitologist, there's no good parasitologist who's worth his salt who doesn't deal with doo-doo. We collect feces, whole feces, which often surprises the community from individuals in the morning. Those stools are set up for a Cato Cats exam. That's a microscopic examination to look for parasite eggs. And all of the positives then are liquefied, make them into diarrhea sort of, and then screen them through a series of sieves, finally through a filter bag here that has a size that's smaller than the diameter of the parasite eggs. It's still not really pure, as you can see, but it doesn't smell really. At this point, we sediment the remainder of the stuff and take the last 5 mLs from the sediment, because parasite eggs are the densest thing in all of this, and they'll be at the bottom. This is, we use only local people to do, well, most of this is all people from the community. I have always been amazed, no matter where I've been, in Kenya, in Brazil, in Egypt, that local people, maybe rural, urban people that live in slums, they're really capable people. New Orleans, they're really capable people everywhere you go. And in fact, the people that I work with are sometimes really amazed at what they see. And I heard somebody comment, this was very gratifying, therefore, for the grace of God go I, because they recognize that people are just as competent as you will see almost anywhere. In any case, this is false. Normally I'm over here doing, working on the sieves, but we do examine them later to see what, try to count the eggs afterwards. And this is the result of about 8 years of repeated therapies in these two communities. Start off with a prevalence of 45%, which was relatively high at the time. It's hard to find places that are that high in this part of Brazil. And then have gone down to a prevalence before the last treatment of about 5 to 6%. And by now, when we left, it probably is more like 3%. Usually you have whatever the prevalence is, probably below 3% in both of these places. Another way to look at this is the egg counts. That is, because we do whole stools and we measure the number of eggs per gram of stool, we weigh the stools, we can give you a total number of eggs. So the total number of eggs that were available for the environment when we started was around 3 million per day. And you can see that we're down to less than 100,000 eggs that are going to be available in the community by the time we finish. So we really should have made a huge impact on transmission by more than decimating the parasite population here. So what are the things that we discover by doing the genotyping and doing the population genetics? And I won't bore you with the genotyping. Actually, it's not boring, but you may not be interested, and it will take a long time to explain it all. But the analysis that we make out of the genotyping we do out of the parasites from each individual are the following. One is that we looked at the day-to-day difference in the genotypes, first we looked at the sample-to-sample difference. That is, we take the same sample and we analyze it twice for differentiation, and that gives us 0. That should be what the minimum that we should find. And we find that to be 0.007 is essentially 0. The next thing was to look day to day. We do 3 stools, but maybe Mrs. Schistosome has a headache on Tuesday and doesn't want to lay those particular eggs. Maybe there are genetic differences from day to day. And we looked at that and we found that the differentiation is 0.01. It's really not significant differentiation from day to day. The benefit from that is that although people find it, believe it or not, well, I guess you believe it, it's really bothersome to come 3 times and ask for your stool. If we only get 1, that probably is representative for the entire week. So that was actually something important to know, because sometimes we don't get all 3 stools. Between individuals, we found that the differentiation was 0.8083 or 0.122, depending Gini Papo and Volta do Rio. The significance is that between any 2 individuals, the parasite population, and it is a population that each individual carries around with them, is different. That each individual is getting a different sample from the environment of these genotypes, and no one individual actually encompasses all of the genotypes that we find within the community. It's all the more reason to sample widely and to get a large sample in order to be able to see what the variation really is between these organisms, between infections in different people. Between different host types, that is, are there different parasites in women and different parasites in men? And perhaps there are some genotypes that like to infect kids. No. The parasites really don't care. They are opportunists. If you are in the water, they will infect you. They really don't care. They have no prejudice against anybody, which is actually useful because we don't have to wrap our minds around differentiation based on these demographic characteristics. Between these two communities, these are communities separated by 6 kilometers. Everybody knows everybody in these two communities. They have relatives in both places. We found a differentiation in the whole population, the communities, of 0.045. And that's not exactly 0.05, but it does show that there is a block to gene flow between these two communities, which this is becoming a mantra with me. It's what we learned from COVID has nothing to do, well, little to do with the sequence along 30,000 stupid nucleotides. It has more to do with looking at how we behave more than anything else is what it tells us. And what this tells us is that, we already knew this, that people don't do their duty in somebody else's backyard. So in the morning, they're not gonna have their bowel movement 6 kilometers up the street, that people tend to stay close to home with that. It also tells us that the local school, where all the kids from Volta do Rio come down to Genipapo to go to school, that the toilet there must be working very well and they must have a septic tank there, because otherwise we would see these two parasite communities begin to merge and look very much the same. I should comment that again, this is a middle income country, which means that at least in this area and many rural areas in Brazil, everybody has running water. They may not drink the stuff, but running water in the house. And that running water often supplies the toilet. Everybody has a toilet. 100% of people have a toilet. But that toilet, in 60% of the cases, flushes to the river. It does not flush to a septic tank. So there is plenty of stool that gets distributed within the water, within the river, that allows the parasite to then multiply and infect snails. So reinfection is another issue that some people feel that maybe we can't treat these things and eliminate them because praziquantel only puts the worms to sleep. They wake up all of a sudden and they start laying eggs, that there's not really reinfection. And what we can look at in terms of the differentiation is what infected you last year and what infects you this year. Are those parasite populations the same? If they're the same, then the parasites just woke up and started laying eggs again. If they're different, this represents reinfection, that you got a new set of parasites. What we found is that there is strong differentiation between the parasites, especially the longer you go over time, that people are reinfected by these parasites. And finally, with immigration, there are some measures of immigration, genetic measures. They're not very good, but we know that at most it's 2 to 3 people a year that will transit from one community to the other in terms of their residence. So it's not a lot of immigration that happens by people, which means there's not a lot of immigration by parasites. Another issue is resistance, and resistance is actually looking for selection. That is, if we have a selective sweep, that all of a sudden we walk in and we get rid of all the parasites that are sensitive to praziquantel and leave the ones that are not, those parasites are going to be intrinsically different. They represent a different population than the population of parasites that were sensitive. And we can look for that. So that if we start off before treatment with this diversity of parasites, treat them with praziquantel, the differentiation, even though the numbers are small, the differentiation should be small between pre and post if there is no selection. If there is selection, on the other hand, the differentiation between the starting sensitive parasites and our resistant population should be different. So the D should be large. The differentiation between them should be large. And the answer is we have 0.007 in Genipapo and 0.009 in Volta do Rio between years, which is to say this is essentially zero, that there's no differentiation between persistent parasites and the sensitive parasites. How is that explained? I think, as we talked earlier, that if you are rapidly reinfected, less than 4 weeks before you got the drug, then you will have parasites that persist. And those will be parasites that are from the sensitive pool. And that probably is the explanation for why we had persistence. The other thing is that we only had a reasonable number of persistence in the first year. After that, we hardly ever saw persistence, which is a pretty good indication as well that there isn't really resistance to this drug in this population. The golden value that I mentioned of the effective population size was measured between just a few of these years. The starting value was 33,000 as an effective population size, which isn't the real population size. It's only as far as my computer could compute it. It's much greater than that. By 2012, it was 2,000. By 2013, it's about 1,000. Over this time, we did not do a study in 2013, but all the way to 2012, we already had a fall to about 1,000. And sorry, this is Genipapo on this side and Volta do Rio on this side. So we're already down to 1,000. We more than decimated the population of parasites. There are some studies that suggest that effective population size less than 5,000 or less than 500 will lead to the eventual collapse of a population on its own. Those are on populations that probably don't represent parasite populations. We don't know what the number is for Genipapo and Volta do Rio. And in fact, since 2017, I have not had cash to go back. But as soon as I am able to, we would like to go back and examine the population today and see where we're at after about 5 years of non-treatment, what the impact has been on that, given that we had an effective population size in 2012 of 1,000 and probably, we'll calculate it later, 2017, much less than that. Another thing that we do, I've been working at this for 30 years, but it's odd that I never really emphasized, although I knew, that schistosomiasis is a disease of human fecal contamination of surface waters. So we finally started to look at surface waters in Genipapo for exactly that, human fecal contamination. There are specific markers for human feces, that is some bacteroides that are not exactly that are human indicative and more than they are for cows or other things. And in a rural area, if you're looking for E. coli, you're going to find E. coli, but you never know where it's coming from. So we're using markers that are more specific for humans because, as you can see, there are all kinds of animals there. In addition, this also stimulates the interest of the population. As you may notice, we have somebody observing us, being very careful, wearing boots, having gloves, using this pole to get a water sample, which is a very curious thing to do, and observing what we do. What we found with that analysis, however, is that if you look at the human distribution, and this is a heat map, the red is where the highest distribution is, the green is the lowest, along the Jiquilissa River in Genipapo. We see that they kind of conform to the intensity of infection with schistosomiasis. It's not surprising. The more people, the more infections you have. This is in 2009. In 2012, although there may be some indication of an increase here in this lower part of the river, it seems even clearer in 2012 when there's a 24% prevalence of the parasite, that we have infections in the lower parts of this river compared to almost anywhere else within the community. And also on the southern side of the river, which I must point out is on the opposite side from the highway and the opposite side from the school. So everybody has to cross this stream in order to go to school, and everybody crosses this stream in order to get to the highway. Some people use logs that are thrown across there, but generally, sometimes the river is up to your waist, but generally the river is wadable, and most people just walk through the river. By 2013, what we find is an even clearer concentration of parasites in the lower end of the river. This, by coincidence, or perhaps not by coincidence, understandably, is also where we have the highest concentration of human fecal contamination, is towards this end of the river. Not because people are so nasty in this end of the river, but they actually receive all of the feces from upstream down to this part of the river. So all of this is consistent with what we see in the community. So the distribution, that's in Portuguese. So the distribution has a correlation with the concentration of fecal human bacteria in the river, and with correlation with the necessity to cross the river. And in case you're wondering, the gentleman here in the lower part of this did step in the river in his very next step. We also talked to people, of course, and we presented all of this to people, and we asked, well, you guys are you still swimming in this river here? He said, no, since you guys have been here, this part of the river is way too nasty. We have a place upstream that everybody goes to swim. Finally, there is one thing that I'd like to hit just as quickly as possible, is that schistosomiasis in Brazil is not confined to rural areas, although principally it's thought of and principally it's focused on rural areas. There is urban schistosomiasis. And the question is the source of urban schistosomiasis, because that'll have an impact on what we do in public health. Once again, this is Brazil, Bahia, and here is an inset on this map is the city of Salvador, which is on a peninsula around the largest bay in Brazil, All Saints Bay. If we look at the population growth in Brazil, what you can see here is that the urban population since the 1960s has totally taken off and dominated. Brazil today is more urban than the United States. It's 88% urban. In the city of Salvador, compared to Brazil as a whole, the population has increased even more rapidly in this city. This is a cartoon that I like from the local paper. It says, "Howdy, my name is Chaos. Glad to meet you. My name is Salvador." I don't know. I like this better than most people, I guess. This is Salvador. It's a place that has grown 300% in the last 30 years, which is a growth that outpaces almost any place in the United States, and certainly more than almost any urban area could really deal with effectively. And so there is a lot of informal housing. In fact, there's mostly informal housing. And although there is access to sanitation, it says that 96% of the city is within reach of the municipal sewer system. Very few people connect themselves to the sewer system. Oddly enough, even though they pay for sewage, that is, everybody gets piped water, everybody has a flush toilet, but many times those toilets flush to rivulets around their houses, and they haven't put in the connections to the sewer. So there are a lot of streams that are set effectively open sewers. That's not to say, don't get me wrong, Salvador is a touristy city. It's New Orleans of Brazil. People go there to party. You go there, you won't get schistosomiasis. It's a very good, nice place. But there are a lot of neighborhoods in which sanitation is not what it should be. I've concluded already? Yes. So let me back up a little bit. What we concluded was that it's possible that all of this, the schistosomiasis we see there, is due to immigration, because the city's grown so quickly. People come from rural areas, and those areas are areas that are known to have schistosomiasis. Maybe they walked in with schistosomiasis. The other possibility is that it's transmitted locally. And the response to public health to those two situations is very different. If it's not being transmitted locally, you just want to identify the people that are infected, get them treated, and the problem is resolved. If it is being transmitted locally, you have to do more in terms of water contact, in terms of increasing the amount of sanitation that you can provide in those neighborhoods, and to treat everybody who happens to be infected. And what we find is that there is very little, by genetic markers, very little evidence of immigration. Almost all of this looks like local transmission, very much like the case in Genipapo. If we could have resolved it in each little neighborhood in which these islands of schistosomiasis exist in urban areas, it is not likely that they would rapidly be reinfected and develop infections again by immigration. That tends not to be the case. Immigration is probably what starts all this, but it takes a lot in order to get it started. And once it's started, it's very hard to stop. Conclusions: populations, infrapopulations, and component populations. I haven't defined those for you, but different areas of grouping the population of schistosomes show moderate differentiation over short distances. The difference from Genipapo to Volta do Rio, 6 kilometers, we already see that there's differentiation between them. So people coming from very distant places, we expect more differentiation as a way of indicating immigration. There is moderate infrapopulation differentiation. Infrapopulation is the infected in one individual. That is, between two individuals, their populations are differentiated. Each individual gets a different group of parasites from the environment. Urban schistosomiasis is an emerging problem with increasing urbanization and poor sanitation. That transmission does occur in cities. And that local transmission is likely responsible for most of the transmission in rural and urban areas. This has public health implications, as I mentioned. The implications for Bahia are that migration contributes less than local transmission, that the distribution is fragmented, that resistance is not easily developed. Treatment across a broad segment of those infected combined with community-wide mass drug administration is what is recommended, and structural modifications to reduce risk for water contact and increase sanitation, which is something is more possible in urban areas than actually rural areas. Thank you. And I'll give this back to Dylan.
Yeah, we actually have a question from Victoria. Did this project require any approval from the state or municipal government?
Victoria, you know it did. It required approval at the IRB from, at that time I was at Case Western, and what we do today from Tulane. It required approval at the local IRB in Brazil. And because it is an international project, it required approval at the national IRB in Brazil. What they're trying to do is avoid abuse by people throwing a whole lot of money at a researcher in Brazil to do studies that are probably not useful for the country and perhaps unethical, so that there are two levels of review that occur in Brazil. In addition to that, one also has to sign off on the removal of biological material from Brazil for their genetic patrimony. And part of that is because we have a lot of Brazilians who do some of these studies in our laboratory.
We have a few more questions. One from Jarna. I'm sorry if I'm saying that incorrectly. Could the water be treated to eradicate the parasite?
Not exactly. There is an approach. I mean, realize that that's why you should ask these questions, because it is a good question. But you just have to turn it around a little bit. At the tail end, when you're very near eradication or elimination of the parasite, because you're not going to do it with treatment alone, some people will treat the water for the snails. They'll use molluscicides to kill snails. The only problem with that is it's not very effective if there's a lot of transmission. Snails are hermaphroditic, these snails, so that you have to get every single snail, or else they're going to be back in 30 days to the same prevalence that you had before. And the treatment for the snails also kills a lot of other invertebrates, as well as it kills fish. So it's actually not, in some places you could do it just by good judgment, but in Brazil you would have to have approval from the Brazilian EPA in order to be able to do that, because it has a strong environmental impact. The parasite itself is only in the water for maybe, it's there every day, but for 8 hours. And if it's running water, it would be very difficult to treat surface water that way. Water that comes into the house, one, is not really a source of infection, and two, is usually treated anyway with chlorine. But it's really not the main source of transmission. Other ways that people have thought about doing this is, can you prevent the parasite from penetrating through the skin? So give everybody rubber boots. If you work out on the water in a tropical place, people don't tolerate that. It gets very hot and you end up with athlete's foot. In addition, your foot gets stuck in the mud. No, that hasn't worked. People have also thought of putting on salves that would prevent the parasites from penetrating through the skin. But that also hasn't been very effective, or has not been able to get a population interested in that.
Thank you. We have another question. Do you have any preference between working with government agencies, multilaterals, or local health systems?
The first and the last. That is, we always work, it's not exactly the government, but certainly with universities. But ultimately, even the universities work with the health system. And in Brazil, there is a national health department, there is a state health department, and there is a municipal health department in every municipality. So in Salvador, we have to work with we don't have to, we work with the municipal department of health. And when we go to Ubaíra, the village, Genipapo, we work with the local department of health. It has huge advantages. One, the population is, people trust this part of government in Brazil. That's not always true everywhere I've worked. Some places people don't trust government. But in Brazil, the Oswaldo Cruz Foundation and many of the departments of health are trustworthy. So you get that as trust. In Genipapo, the Department of Health gave us a laboratory to work in Genipapo. It's in the old clinic that they had there. And they fixed everything up. They put in new faucets. So we get a lot of cooperation from working with them. And what we do, our results go directly to those departments, which are the people that will first be able to, well, second be able to use them. First people are actually the community itself. But that's why we like to work with departments of health. It's good to work with universities as well, because part of this is also technology transfer and developing capacity. Brazil has a lot of capacity. They sometimes give us advice about how to do our genotyping, but still there is a lot that we can provide for them in terms of training that they don't get elsewhere. That wouldn't happen if it was just us working. And it's the most satisfying way to work.
Our next question is from Alyssa. Are you investigating genetic differences between urban and rural areas to determine the source of infections? And if infections are immigrant sourced, is it possible that schistosomiasis will be established locally in urban areas?
Yeah, that's exactly the question in both areas. That is, does immigration is a reason for persistence in a rural area, for instance, which we didn't see, or is it the reason for presence and persistence in an urban area, which also we didn't see? I didn't give you the numbers. We didn't have a lot of time. But if you did a comparison between, as it turns out, we had a sample from 2007 and 2011, and we compared those two of a whole population, and we found that the D was 0.007. That is, there was no differentiation over those 4 years. There had been a lot of immigration. We're going to see a differentiation between them, and very little was seen. In addition, there are other things that I don't have time to point out, but there are other ways that we can indicate that most of this is local transmission and not from immigration. Although, like I said, I think sometimes it is established by immigration, but it's not maintained by immigration. And it's not easy to establish by immigration.
Our next question is from Rosemary. She mentioned she had to log off of the call for a second, so she may have missed it, but what are your feelings on predatory biologic control?
It's not a feeling. You could ask for the evidence. People have used biologic control, tried to use biological control in many places, Africa in particular. Should be popular this time of year in New Orleans. Crayfish have been one mechanism for trying to control snails, because they will eat snails, but they also eat small fish, and people were not entirely happy with that. There are predatory shrimp in Senegal that have been tried, but those have not been easy to establish. They were there naturally, but got removed when people did some of the dams on the Senegal River. People have been trying to reestablish them, but it's not really been successful. People have tried ducks in Japan for japonicum, but a lot of those ducks ended up in pots on Sundays, and didn't seem to ultimately be a good way to control it. In St. Lucia, some people swear by predatory snails, that there are snails that will crowd out the snails, not predatory, that will crowd out the snails that transmit schistosomiasis, a very specific species. But that is not entirely clear that that actually happened, and nobody's been trying to do that. It's always dangerous to introduce foreign species into a different area, and nobody would be anxious to do that in Brazil. One species of snail that was introduced for escargot, it was an African snail that got released and has become a pest in there, everywhere in urban areas. In fact, there are these huge snails that crawl around and slime up the entire steps in the city of Salvador because it gets stepped on all the time. So there are risks with doing that. People are still examining, but no one's found the magic bullet for biological control of snails.
This next one isn't a question, but Victoria wanted to let you know that your rotating globe is very cool. I think we can see it behind you.
Yeah, I like that. I gave a presentation at the, I don't know, the CWRU Model UN, And they made it out to Dr. Robert E. Blanton. I've always loved that. I'm not a Robert E. Blanton, but I have to admit I like the globe as well. It's solar. So.
Very cool. Well, thank you so much, Dr. Blanton. I really appreciate you being a part of our celebration and programming for National Public Health Week. And thank you to everyone who watched the presentation today. If you go to our website, you can view all of the further events happening during our National Public Health Week programming. We've got some more presentations throughout the rest of the week and a really cool leadership panel happening on Friday. I will put my email in the chat, but once again, thanks to all of you for joining us.
Perhaps you can tell I had a good time too.
Yes, we can.
Thank you.
Thank you again.